Good evening, everyone. Good evening, everyone. How are you all? Very good evening, Siddesh. Good evening, Shraddha. Good evening, everyone. Good evening, Soumya Jyoti. Okay, so my name is Muskan Kashyap, and I'm a bioinformatics analyst and the founder of Biotechshalaa. So I will be your mentor throughout this entire program. Okay. So before starting today's session, I want all of you to understand one thing very clearly. Okay, listen to this very carefully. First thing is that you do not need any prior knowledge. Okay, we are talking about that you do not need any prior knowledge of coding or programming to start learning bioinformatics. And second thing, you do not need to specifically come from a biotechnology or bioinformatics background. Okay, because bioinformatics today is used in almost every area. Bioinformatics is used in microbiology. Bioinformatics is used in genetics. It is used in plant science. You must have heard of the word cancer research. You must have heard of forensic science, vaccine development. So bioinformatics is used there as well. If we talk about zoology, if we talk about agriculture, if we talk about drug discovery, personalized medicine, evolution studies, and even AI-based biology research. So bioinformatics is used in all of these areas. So regardless of your background, this field, bioinformatics is useful for you. Okay. And this program is completely beginner friendly. So if you feel that you do not know anything right now, so that is completely okay. Today, we will start from the absolute basics. So are we clear with these things? Let me know in the chat box, are we clear with these things? Okay, very good everyone. So first of all, let's understand why bioinformatics was created. Okay, we will understand this question, why bioinformatics was created. So we will take an example to understand this question. What you are currently looking at the screen is the human genome. Okay, what is it? Just imagine that this is a human genome. Okay, now the human genome, it contains around 3 billion base pairs. Now, do we understand the term base pairs? Means DNA has 4 bases, A, T, G and C. So A pair up with T, so that is one base pair. G pair up with C, and that is another base pair. Okay, so we are talking about human genome, that the human genome contains around 3 billion base pairs. That means we are talking about 3 billion letters of A, T, G and C. Now think about modern biology. Okay, we are talking about modern biology. So every day scientists generate these DNA sequences. And what are these DNA sequences? These letters A, T, G and C. So in the same way, scientists generate RNA sequence, they generate protein sequence, they generate disease mutation data, drug target data. So now you can imagine the size of these data, right? The human genome alone contains around 3 billion base pairs, and scientists are generating similar type of biological data, not just for humans, but for thousands of organisms. It may include bacteria, it may include plant, animals, and many other different species. So the amount of biological data, it became so huge that biology alone was not enough anymore. So do we understand that biology alone was not enough anymore because the data got so huge? So scientists needed computers, and that is where bioinformatics was created. So what is bioinformatics? Bioinformatics is the field where biology plus computer science plus statistics and data analysis work together. So in simple words, bioinformatics help us to store, to organize, to analyze, to understand biological data using computers. Let's take an example to understand bioinformatics. Okay. For example, just imagine that you are from microbiology background. Okay, it's possible that any one of you can be from microbiology background. So for example, just imagine that you have a degree in BSc or MSc in microbiology. So you are studying microbiology. Okay, and imagine that one day you are working in a clinical laboratory in a hospital. Okay, we are taking an example to understand bioinformatics. Now, you are working in a clinical laboratory in a hospital and you have a degree in microbiology. Now, a patient comes to the hospital and that patient has a bacterial infection. Okay. Now, as a microbiologist, your job is to identify the microorganism which is causing infection to the patient so that the doctor can prescribe the correct treatment. Now, firstly, what you will do as a microbiologist, you will collect a sample from the patient, right? For example, you can collect the sample as blood, urine, or any other relevant sample. Then you will grow the bacteria in the lab and you will study it. Now, as a microbiologist, you want to answer three important questions. Question number one, that which bacteria is this? Question number two, is this bacteria harmful? And question number three, does this bacteria contain antibiotic resistance genes? So to answer these three questions, the DNA of the bacterium can be sequenced. What did I just say? That the DNA of the bacterium can be sequenced, which means the microbiologist can read the DNA sequence of the bacteria. So that is where bioinformatics comes in. The DNA sequence of the bacteria, it can be compared with thousands of bacterial sequences. And these thousands of bacterial sequences are available in the databases. So we will study about these databases as well. But for now, just understand that the data, the biological data of these thousands of bacterial sequences is available in the databases. Now, then using the bioinformatics tools, the microbiologist can identify the bacterial species and they can even detect the genes which are associated with the antibiotic resistance only with the help of bioinformatics tools. So this is how bioinformatics helps microbiologists. So do we understand the point of bioinformatics in the field of microbiology? Did we get any idea here? Very good, Soumya. What about others? Did you got any idea how bioinformatics is we are using bioinformatics in the field of microbiology? Just we are getting an idea today. Okay, today's session is all about introduction. Okay, similarly, let's take another example. Let's take example from plant science. Okay, suppose you are from a plant science or agricultural science background and you are studying rice plants. Okay, now imagine that there are two rice plants growing in the field and there is a drought condition. Okay, there are two rice plants which are growing in the field during a drought condition. Now, one plant survives and it continues to grow while the other plant dies quickly. Now, as a plant scientist, okay, now imagine yourself as a plant scientist. So, as a plant scientist, you want to answer an important question that why one plant is able to survive the drought condition while the other plant is not. So, to find the answer, scientists can sequence the genome of both the plants, which means they can read the DNA sequences of both the plants. And then again, they will use here bioinformatics tools so that scientists can compare the DNA sequences of both the plants and they can identify the genetic differences between them. Now, after the comparison, they may discover specific genes which are associated with drought resistance. So, this information here, that which gene are responsible for drought resistance in a plant, so scientists can develop improved crop varieties that can better survive under drought conditions. So this is one of the example of how bioinformatics can help in plant and agricultural science. Let's take example from genetics. Okay, imagine a child has a genetic disorder. Okay, we all understand what are genetic disorders. So imagine that a child has a genetic disorders which has been running in the family for generations. Okay, now the doctor know that the disease is inherited, but they don't know which gene or which mutation is responsible for it. So, as a geneticist, your goal is to identify that genetic change that is causing the disorder. Now, the scientists, they can analyze the patient's DNA using bioinformatics tools, and they can compare the patient's DNA with the reference genome. Do we understand what a reference genome is? Does anyone here know what a reference genome is? Should I explain what a reference genome is? Okay, so for now, just get an idea that a reference genome, it is actually a standard DNA sequence that the scientists use to compare the another DNA sequences. Okay, so by comparing the patient's DNA with the reference genome, scientists can identify the positions where mutations are present in the DNA. Okay, so what are we doing here? Scientists are comparing the patient DNA with the reference genome. And what is a reference genome? It is a standard DNA sequence which is used to compare with the DNA sequences. So we are comparing the patient's DNA with the reference genome using the bioinformatics tools. And that is how scientists are able to identify the positions where mutations are present in the DNA. So did we get the idea here? How mutations are found out using the bioinformatics tools? For now, we are not learning about any particular type of bioinformatics tools. We are just getting an idea how bioinformatics is used in every different kind of field. Okay. So, once the disease causing mutation is identified, the doctor can better understand the disorder and they can provide more accurate diagnosis and treatments. Okay, so here, bioinformatics here, it helps geneticists study the inherited diseases. Now, let's take example of zoology. Okay, for example, you are from zoology background. So, here, the scientists who study zoology. So scientists often compare the DNA sequences of animals to study the evolution and species relationship. For example, let's take an example. Scientist wants to compare the human DNA with the chimpanzee DNA. So if we are comparing the human DNA with the chimpanzee DNA, that's how we are going to find the evolution, right? That's how evolutionary studies are done. And again, bioinformatics tools are used here. So they calculate the sequence similarity. Okay, to calculate the sequence similarity between the human DNA and the chimpanzee DNA, bioinformatics tools are used here. And again, to study the evolutionary relationship between the species. So zoologists here, they also use bioinformatics. If we talk about pharmacy, so this is a very interesting point about pharmacy here. So before testing a drug in a laboratory, okay, any drug that is being tested in a laboratory, so before testing that drug, scientists, they first test it computationally. Are we understanding this line? That before testing a drug in a lab, scientists, they first test it computationally. Suppose, for example, scientists want to develop a drug. Okay, some scientists want to develop a drug against a disease protein. So there is a protein which is causing a disease. Okay, so scientists want to develop a drug against that disease protein. Now here, scientists will use the bioinformatics tools, they will use molecular docking software, and they will check whether the drug molecule can properly bind to the target protein. So our target is that protein because that protein is causing the disease, and here scientists have developed a drug so that the drug can stop that protein. So here, before testing that drug in the lab, scientists test it computationally. And how they test it computationally? By using the bioinformatics tools and molecular docking softwares. So here, this saves both time and it also saves the laboratory cost. Now let's talk about those students who come from the computer science background. Now, again, let's talk about modern biology. So we understand that modern biology, it generates massive amount of data. Okay, modern biology, it generates massive amount of sequencing data every day. Now, we need someone to build a software. We need someone to build some pipelines. We need someone to build some AI models, to build databases. We need someone to build some visualization tools. So these are the computer science students here that help develop these systems so biological data can be analyzed efficiently. Okay, so now you can understand one thing very clearly, that no matter, okay, no matter which field you belong to, bioinformatics is becoming important everywhere because modern biology is now data driven. Now what do we understand by the term data driven? I just said that modern biology is now data driven. It means that the biological research today, it depends heavily on large amount of data and computer based analysis. So let's understand it more clearly. So earlier, let's go back to the previous times. So earlier, scientists used a method called Sanger sequencing. You must have heard of Sanger sequencing. If you did not heard it, it's okay. So let's just continue with it. So earlier, scientists used a method called Sanger sequencing to read the DNA sequences. Okay, for example, DNA sequence of a human. Now, the Sanger sequencing was a very important discovery in biology, but it had some limitations. So, it was very slow, it was very expensive, and scientists could only analyze small amounts of DNA at a time. So later, newer and much faster sequencing technologies were developed, and these modern technologies are now called next generation sequencing. Okay, these modern technologies are now called next generation sequencing technologies. So some examples of these technologies are Illumina, Iron Torrent, PacBio, Nanopore. So at this stage, okay, at this stage, you do not need to understand how these technologies work internally. Just remember that these are the modern technologies which are used to read the DNA much faster than the Sanger sequencing. So because of these modern sequencing technologies, scientists, they started generating the DNA a lot. Okay, scientists generating massive amount of biological data. So now imagine that if one modern sequencing machine, okay, this is one sequencing machine here. So imagine that if this one sequencing machine is producing millions of DNA reads in one day. So we are talking about that this machine is producing millions of DNA reads in single day. Now, if we are generating millions of data every single day, so how will scientists store that data? Okay, how will scientists store that data? So this is one question. Second question, how will they organize it? Because we are talking in millions. Third question, how will they compare sequences? Because earlier in these examples, what we understood that we are comparing the sequences, right? So if we are generating millions of data in a single day using these modern technologies, so how will the scientists compare the sequences? Another question, how will they identify mutations between these millions of data? How will they find disease related genes? How will they study evolution? Because the data is so huge. So doing all of this manually, it become impossible. Okay, it is almost impossible to do this manually. So scientists, they started using computers and software tools, databases, and bioinformatics algorithms to analyze the biological information. So computational analysis, it became very necessary, and that necessity created the field of bioinformatics, and that is why biology is now called data driven. So do we understand what we have learned just now, or do we have any questions? Are there any questions? What we have just learned now about bioinformatics? Okay, very well. So now, before we use bioinformatics tools, we first must understand that what kind of biological data we are dealing with. Okay, so because by now you must have understood that bioinformatics is mainly about analyzing the biological information. Okay, so there are three important types of biological sequences. So the first is DNA sequence. Okay, so DNA sequence, it contains 4 nucleotides A, T, G, C, as we all know. So this is an example of a DNA sequence. So this is a biological data. So this is the data that we actually deal with. Second data is RNA sequence. So in RNA, there is only one simple difference, that in place of T, we have U. So here U stands for uracil. Okay, so instead of thymine, in RNA, we have U. So if in a sequence you see U in place of T, that means it is an RNA sequence. So this is an example of an RNA sequence, which is also a biological data that we deal with. Third is protein sequence. So proteins are made of amino acids. So proteins here, they are represented using single letter amino acid code. What is it? Single letter amino acid code. For example, here in DNA, it is ATGC. Adenine, which is represented by A, thymine represented by T, guanine represented by G. In the same way, in the protein sequence, these amino acids, these are amino acids, okay? So they are represented using single letter amino acid code. For example, here, this first amino acid is M, which means methionine, and is represented by M, which is a single letter amino acid code. Here we have K amino acid, which means lysine. Okay, it is lysine, which is represented by K. Again, which is a single letter amino acid code. In the same way, we have T, which stands for threonine, represented by T. Again, it is represented as single letter amino acid code. Okay, so in biology, there are 20 amino acids which are used to make proteins. So in bioinformatics, these sequences, okay, these are the sequences that is the data. So DNA is the data, RNA is the data, protein is the data, and our job is to analyze these data. So do we have any questions here? Okay, so are we clear with all the introductory part that we learned today? What is bioinformatics? How bioinformatics was created? What data do we deal with in bioinformatics? And did we get any idea how different fields use bioinformatics? Okay, very good. Shraddha, very good Bhargavi, very good Disha, very good everyone. So today I would like to give you a very small assignment. So the topic of your assignment is applications and limitations of bioinformatics. Now understand one thing very clearly, that I do not want you to simply copy information from ChatGPT or Google or any website. Okay, I do not want that. I want this assignment to be research oriented. I want you to do some research how bioinformatics is practically applied in different areas. Okay, try to explore how bioinformatics is actually being used in the real world. For example, you can look at the applications of bioinformatics in disease diagnosis. If you are interested in drug discovery, you can look there as well. If, for example, if you are interested in agriculture or genetics or microbiology, so any other area that interests you, so do your research how bioinformatics is used there. Okay, similarly with the limitation part as well. So you can think about, for example, is bioinformatics always 100% accurate? If not, that means there is some limitation. Okay, so are we clear with the assignment that it should not be theory, it should not be copy paste, it should be research oriented? Okay, again, I will say to you again, the goal of this assignment is not just to complete homework, but to start thinking like a researcher and understand how bioinformatics is applied in real biological problems. So are we clear with the assignment? Okay, so you will submit this assignment by Sunday on Google Classroom. Okay, I will upload this announcement on the Google Classroom, and by Sunday you will upload your assignment. Okay, now everyone kindly raise your hand so that I can take your attendance. I will be taking screenshots, so make sure that you are raising your hands. Okay, some students are still not raising their hands. Okay, so I have taken your attendance. If you have any doubts or any questions you want to ask, you can ask me. Otherwise, you may leave today's class. Okay. So today's was all about introduction. If you still have any problem in the introduction part, you can text me. Brendan, the time is the same, 7 to 8 p.m. Indian Standard Time. Okay, Brendan? And the class link will be shared 5 minutes earlier. Darpan, it depends on you. It doesn't matter that you have to specifically type everything, but it should be practical, like you should gain some knowledge from this assignment. Okay, it should not be just theory and copy paste. Okay, I want you to do your own research so that I can see in your assignment that what research you did. It should show that what you learned. Okay. Okay, everyone, then thank you for joining today's session. Okay, good evening, everyone.